Yeah, so hello everybody who is hearing me. I'm greeting you from the University of Freiburg in a little town in southern Germany, close to the Black Forest, which has a long tradition. My university has a long tradition in studying the immune system, particularly B lymphocytes, because the reason why I'm here is that Georges Köhler, the Nobel Prize winner, with Cesar Milstein, who discovered the way how to make monoclonal antibodies, was the director at the Max Planck Institute here. And now I'm here professor of molecular immunology. So now I need my presentation. Okay, so I want to discuss with you. First, I want to thank IOAIS to give me the possibility to present you our favorite system right now, namely the Ramos B cell system. And I will tell you how we are using this system also to learn something about humoral immunity against anti-SARS-CoV-2. So my story actually doesn't start in Freiburg, but in Houston. You all know that Houston is the center of the USA space exercise and space exploration. And on the April 13th of 1970, Apollo 13 was on the way to the moon, where suddenly an explosion happened in the gasoline tank. And that was a big problem for Houston. But they luckily got the whole crew down back over the moon, via the moon, back to the Earth. And so Vidal mentions this. There was another problem in Houston happening a few months later. And it's a sad story of a young child, a boy, who developed the Burkitt lymphoma and was admitted to the St. Joseph Hospital in Houston. And unfortunately, at that time, the clinicians did not know how to save the life of this young child, but scientists were racing to understand more about this tumor. And there was a particular fortunate situation that in Houston, there was a very well-funded science institute, the Baylor College of Houston, which had outstanding scientists, and they tried to learn more about these tumors. So they managed to get cells from this little boy in culture and start to study it. And this is an outstanding group of scientists at that time in the laboratory of David Mumford. The work was mostly done by Peppino Giannovella. And it happened by chance that George Klein, one of the outstanding tumor immunologists, made a sabbatical in the US at the time, and he knew about their studies, so he helped them in developing the system. And importantly, they found out that this Burkitt lymphoma was not generated like other Burkitt lymphoma by Epstein-Barr virus transfection, so it was a negative line, and they could test the virus spread with the cell system. So since then, since 1972, Ramos cells grow in culture and can be used for multiple studies all over the world. So the Burkitt, as we now know, Burkitt lymphoma is a B-cell tumor, which is derived from a very critical area, the germinal center region, where B cells are selected for high affinity antibody production and memory B cell generation. And out of this area, where there is also somatic mutation happening, tumors can develop, and one of them is the Burkitt lymphoma. When you look in the internet up to learn more about the St. Joseph Hospital, actually you find only very few pictures, and one is this here from 1952. Myself was just 2 years old at that time. And you see that there was another health crisis in the USA at that time. And what you see here outside the hospital is an iron lung to support the breathing of victims, mostly young children with polio. And Yeah, so at that time, polio was a big problem in the US in the 50s. And many scientists were engaged to find out more about this. And luckily, scientists were successful to develop a vaccine which could finish this epidemic. Then over the last part of the 50s and the early 60s. And these two scientists, to mention, the first one is Salk, and the second one is Sabin, who made these two vaccines. So Jonas Salk was the first one who developed an inactivated virus vaccine. And you see that many children were happy to get that vaccine because polio was an awful disease. And there was a big scare for all the parents of that time that their children would caught it. And one has to mention another person here prominently. Without her, these children would have not been safe from this disease, and that is Henrietta Lacks, which unfortunately died in young age on a tumor. But her tumor, similar to Ramos, were put in culture, and it was the first human cell line. And this was a very important source for the expansion of the poliovirus in this human cell culture, which is now called HeLa. And without Henrietta Lacks and without the HeLa cells, the vaccine would have not been able to develop. Shows you how human cell cultures and cell lines can be important. And we think that also Ramos cell could play a role in the ongoing pandemic, and that is one of my topics. So we are living, of course, now in an information age, and we are living on the age of omics. And so these cell lines, which can grow in culture, still provide us with a lot of information because we have their whole transcriptome, we have the whole proteome, we have the whole surfaceome, which we actually generated with, in collaboration with Bernd Wollscheid of the University of Zurich. And we have also the metabolome. So we have a lot of knowledge about these cell lines. And so we know a little bit what drives the growth of these cells. They have a high expression of an oncogenic form of MYC. They have TCL1 driving PI3 kinase activation. They have a high expression of the signaling subunits of the beta receptor, 79A, CD79B, also called Ig alpha, Ig beta, which my group studied since more than 30 years. And particular Ig beta, we know, is expressed as a homodimer on these cells and drives the tumor growth. And we have now another genetic tool with the CRISPR-Cas9 invention. So that puts us in a place that we can now not only better understand these human tumor cells, but we can also modify it in a way this was not possible just a few years ago. So we thus generated not only one knockout line, but many. And one knockout we did is we removed all 4 genes or rendered it defective of coding for the heavy chain, the light chain of the membrane-bound immunoglobulin, and the Ig alpha, Ig beta. So 4 knockouts resulted then in the BCR null line. And we could have studied the difference between the wild-type and null line that we did together with Julia Yelozova's group, who is an expert in metabolic, studied in B cells. And she found out that one reason actually why B cells do not survive very well when they lose a beta receptor is not necessarily only because they lose the tonic signal, but already in the R, where when the beta receptor is formed, there are a lot of signaling going on, which is important for the metabolic fitness of the B cells. So without a beta receptor, the R is much smaller, and the mitochondria doesn't get supported by the R in their metabolic function. And this is a discovery described in the recent published paper from Julia Yelozova with my group. Okay, so what we can also do, of course, we can change this surface completely. We can knock out many things, and we can also express things. And one thing which is just in the current situation important is to learn more about viral proteins of the SARS-CoV-2 virus. And so we have a Ramos cells which we got from Jürgen Wienands and Nicolas Engels from the University of Göttingen, who has an echo receptor. So we can use retroviral vectors to transfect genes into these cells after the knockout or before, and then we can express spike proteins on the cells. Okay, so these spike proteins are prominently expressed on the SARS-CoV-2 virus, which is running around widely right now, and we try to contain it, and we try to find a medication against it and a vaccine. And this is a big issue in many laboratories. I'm actually giving every year in my function as an immunology professor of the University of Freiburg since 2000 already a lecture on the Spanish flu, and told the students how epidemic can be awful for a whole generation. And they always looked at me with doubts. And I showed them this historical picture where all these people looks a little bit strange, and that it would affect their own generation of young students, which have to cover their nose and their mouth. Nobody was foreseeing for a long time, but this is this is now the case of the COVID-19 disease and the pandemic we are living through. Actually, there were early warnings that that the race between the human and the virus are not decided. So a prominent person here to mention is Joshua Lederberg, who lived through the HIV epidemic and worked on this. So he wrote very important made very important statement. He said, humans' only real competition remains the viruses. The survival of the human species is not a preordained evolutionary program. So viruses are pretty dangerous for our survival in this planet. In particular, there are a lot of problems with these SARS-CoV-2, which of course also I, as a molecular immunologist, had a closer look at. So it's a new virus, and there is no immunity, and therefore all the discussion whether SARS is similar to influenza or not, or as severe as influenza, is not really helpful, because we have a basic immunity against influenza. Also, influenza is changing all the time. There is a basic immunity, and there is no immunities against SARS-CoV-2. There may be other coronavirus give us some immunity, and that we can discuss maybe, but there is no major immunity against this new virus. It's an RNA virus. It has a high mutation rate. That's another bad news. And it chooses, unfortunately, a very bad receptor to attack our cells, namely the angiotensin-converting enzyme 2. It's a membrane-bound enzyme, and that happens to be prominently expressed in our lungs. And here's a study, a single-cell transcriptome study of human cells for the ACE2 expression. You see it's prominently expressed in the lung, seen in red, in the heart, but also in the intestine, in the bladder, and to a certain part also in the liver and in the stomach, apart from, of course, the nasal and mucosal and the respiratory tract, where it first attacks our body. So that is bad news, that this target is so prominent in one of the most sensitive organs we have in our body. Another bad news is that the binding site for AC2, when you compare the sequence of SARS-2 against other coronaviruses, then you find there is a mutation. It's an ACE2 binding site, and that makes its binding even high affinity in comparison to SARS-CoV-1, which had a lower affinity to this AC2. And another bad news is that this virus generated a new sequence just between two domains of the spike protein. which have to be cleaved, and it generates a furin cleavage site, so it can be very efficiently cleaved. So here you see a model of the spike protein comprising two roughly domains. The S1 domain and the S2 domains. It's the trimer, as you see. And when it sees its target, the AC2 binding site, the binding domain of this S1 domain can open and bind to it, in particular in high affinity if you are SARS-CoV-2 viruses. And then it is accessible to furin and other proteases. It's not the only one, there are several ones, and that can cleave the S1 domain and then relieve the S2 domain to change its conformation into a needle, which then is the membrane attack complex of the spike protein. And that then results in the fusion of the virus membrane. It's surrounded by a lipid membrane like our own cells. And when the spike is activating its needle, it can fuse these two membranes, the endosomal membrane and the viral membrane, and then release the RNA, the viral RNA in the cytosol, which can then replicate rapidly. So another important statement of Joshua Lederberg is that the future of humanity and microbes likely will unfold as an episode of a suspense thriller that could be titled Our Wits versus Their Genes. So I told you with the first citation said that our survival on this planet, given all the viruses running around, is not a sure thing. The only thing what we can do is maybe set up one organ which we developed quite heavily during our evolution, that is our brain. And therefore scientists all over the world are trying to activate their brains and their skills to learn more about this virus and the disease it is causing. And there's a really amazing mobilization. I find it really exciting time as a scientist. It's a bad time to be a student right now. It's a bad time to live through this, but it's an exciting time for scientists because there is now really a mobilization of the best brains and the best laboratories to learn more about how the virus enters our cells, how it replicates, how it amplifies itself, and how it gets out of the cell, and what is how its interaction with our own body and the immune system in particular. There is some remarkable aspect of this. So the virion is assembled from the spike together with the membrane protein and the envelope protein, and that is not budding out of the plasma membrane, but assembled already in the R and buds into the Golgi. And so one thing we are doing is to apply a system which we are used a lot in the lab over the years, namely a rebuilding system. That was the start of synthetic biology, which we did here in Freiburg prominently, and we have an institute now on this. So one idea was not only to make knockouts and loss of function, but gain of function experiment that you can rebuild a signaling system in this case, right? So we learned a lot about how the B-cell antigen receptor is structured and signals by just transiently express it for a few days in the cells and sees what it's doing. And we learned, for example, that in the resting cells, the B-cell receptor is a dimer, and when antigen comes, it opens up with the help not only of the antigen, but also of this kinase SIG, which is opened in the receptor by an inside-out signaling process. And this is all published. So now we try to apply this system, this established Schneider cell system, in learning how these membrane proteins of the SARS-CoV-2 are interacting, so we can co-express proteins easily in the system. We express a spike, the membrane protein, which we can see in the microscope because it's tagged to GFP, and the envelope protein. So what we learned, for example, when you express the membrane only, then it's mostly stuck in the R of these cells. GFP as a control is all over in the cells. This is mostly stuck in the R. When you express all three membrane-associated proteins, the spike and the envelope, then you see it moves into the Golgi or structures between the R and the Golgi. What we also notice when we look now with a FACS scan or on the surface, that the spike protein alone can actually come to the surface to a certain extent. But when you co-express it with the other membrane protein, it's mostly stuck inside the Golgi compartment because, of course, other essential elements of this virus, namely the nuclear proteins and the genome, is missing. So viruses are never produced. But so you can retain it inside the ER. So that is playing around with viral proteins and learning a little bit how they're interacting and maybe finding drugs how to interfere with this. So by learning that the spike alone cannot surface, of course, brought us to the idea that, hey, why we not express the spike on this Ramos B cells, which is another important cell line we are studying a lot of cells. So the advantage of these cells, as I told you, is they have an EcoR receptor. They can be transfected with retroviral vectors. We use an iris GFP vector so that all green cells are marked, they express the spike. And then we can use a well-known technique for immunologists, namely the flow cytometry, the FACS scan developed by Len Herzenberg and with Lee together. And you see one of the earlier models of this machine, and then can measure the expression. So what we are doing is we take our Ramos cells, we know they express the spike, and here we expose them to 3 sera and test whether these sera contain antibody against the spike. So the spike is expressed on Ramos cells, and the Ramos cells we express the site are GFP positive, so we can stain for GFP, the green color, and we can study the antibody bound to the spike by PA color. And you see that here are 3 sera below, which are clearly positive and express, bind the spike, and we have an easy assay for these antibodies. And there are 3 serum which are negative. There's one interesting sera which has a high binding to the Ramos cells. We do not know why. That is interesting maybe to find out one time, but this is not the focus of our study. So our focus was to get an easy assay for anti-spike antibodies. And this assay, as you all know, can be done in 1 to 3 hours, and you know then the result, whether the serum contains spikes or not. You also can use this assay to learn how monoclonal antibodies, which are generated all over the world against viral proteins, are working. And one set of antibodies we got from the laboratory of Hans-Martin Jeck. He generates, as he told you, at an earlier IOIS seminar series, that he generating monoclonal antibodies against viral protein. We got here the supernatant of 6 of his hybridomas, and you see these all express antibodies binding the spike, so monoclonal antibodies against the spike. And this one is actually also doing it, but it doesn't have a kappa light chain, but a lambda light chain. So you can also measure monoclonal antibody binding, and you can, of course, also, measure also at one point whether they bind to which part of the spike they're binding. So you can do epitope mapping, which will be important for choosing right antibody, monoclonal antibody collections for the treatment of maybe SARS-CoV-2 patients, which can be treated by passive immunization, as Hans-Martin Jeck explained to you. But the nice thing about our assay is it's easy, and we have the cells. And another thing, you do not have to purify the proteins. The Ramos cells produce them on their own, and it's a human cell line which is doing all its job, and including the glycosylation. You see here a picture of the spike. Told you it's a trimer, and you see it's not only these red areas, it has a lot of green areas, and these green areas are sugars. It's a heavily glycosylated protein, and will be interesting to figure out what the role of these glyco groups are in the recognition of the immune system. So we can modify it. We can, as I told you, we can knock out any gene in this Ramos cells. We can target certain glycosylation modifying enzymes. and maybe modifying it in this way, we can express mutant versions of the spike, which is lacking certain glycogroups. So that is one aspect we are interested in, and that can be done in this spike. And of course, you can also not only express the spike of SARS-CoV-2, there are different coronaviruses. Before SARS-CoV-2 came, there was an epidemic, a short epidemic of the SARS-CoV-1, and we have the genome of this, and there was a MERS outbreak. If both of them were luckily confined, and we can test cross-reactivity of antibodies against these spikes as well as to common cold corona spikes, which we now are in the process to clone the genes and express it in our Ramos cell system. Yeah, so that is the way we want to go. We want to express this, and then can easily test in a cytoflow experiment these reactivity of antibodies. What you also can do, and what we did already in the past, you do not delete all genes of the B-cell antigen receptor complex. You only delete the heavy and light chain of the membrane-bound immunoglobulin, and you keep the signaling subunits of the B-cell receptor. And you combine this also with the knockout of AID, that is the enzyme which makes a mutation in the germinal center. It's an activation-induced deaminase, as you all know. So we do not want, of course, when we express immunoglobulins in these cells, that they get mutated. So we knock this enzyme also out. And so you can express other beta receptors. Now, so we replace the Ramos beta receptor with that which of another specificity or interesting, because the one problem of Ramos is we do not know what this receptor is binding, right? The binding activity of this Ramos cells is not clear. So we removed the heavy and light chains and replaced it with bona fide heavy and light chain encoding here, for example, for HENEC lysozyme. And then we can use these cells to test antigen reactivity. And this is very important because, you know, antigens interact with the B-cell receptor in a very profound different way as anti-receptor antibodies. There are many people who think they are doing the same thing. They're not. They're very different. And that we find out in this assay. So we have done it, and you can express different isotypes. For example, IgM, IgD. You know, IgD is a major receptor of mature B cells. Most people do not know that there exists IgD. They don't care about it, but it's the most prominent receptors of more mature B cells. And it actually sees antigen in a very different way. And that you can learn from a publication which we did together. Oh, there is a slide missing. So a publication we did together with Hassan Yuma, which shows that IgM, IgD has a different reactivity to particular forms of antigens. Okay, so what we will do next, and these are also experiments we are planning, is not to study HENEC lysozyme, but study reactivity against SARS-CoV-2. So we can, as soon as monoclonal antibodies are available, and they are available now in many laboratories, we will know the VH-VL combination binding certain epitopes in the spike or other viral proteins. We can generate virus-specific B-cell receptor and then test the sensing of B cells and how B cells and the B cell surface is interacting with either spike protein isolation or with viral spike proteins on the virus. And there is, of course, also pseudoviruses now available which express the spike. You do not have to do this under S3 condition. You can also do it on normal condition by using pseudoviruses and test how the receptor interacts with the surface. And then we have many tricks in this system. For example, we can combine antigen binding also with imaging. So we cloned VR Dennis Saltugokul in my lab. She cloned a snap tag in front of the heavy chain, membrane-bound heavy chain, and now she can stain the cells. These are Ramos cells. I can unfortunately not show the beautiful image. These are Ramos cells in a certain gel, so not squeezing them to awful plastic or a glass surface. So they are inside a nice confinement where they feel happy. And the membrane is stained by GFP CAX, and you can stain the beta receptor by yellow-green color. And we can then follow up how these cells react in a natural environment or quasi-natural environment by a new technique, namely the lattice light sheet microscope, which we study in collaboration with Jakob Pilar's lab in Osnabrück. Okay, so before I want to finish, I want to tell you, of course, we all lived through confinements and to changes in our life. So I'm doing, when I'm at home, doing a lot of yoga. And when I'm doing my yoga, I look at this picture often. And this is the birth of the galaxy from Max Ernst. And for me, it's a symbol for the virus, for the complexity of the virus. I find this picture amazing because every time you look at it, you see other aspects. And that reminds me of the virus. When scientists look at it, we always discover new things. We discover new things about the virus, about the immune system. It shows you the whole complexity, but it also could be a symbol about the outbreak, the viral outbreak, and hopefully the confinement, the end of this outbreak. And that is my hope. And that is why I look every day on this picture. And I hope, of course, that at one point we will have SARS-CoV-2 vaccine. The first time I remember to be vaccinated was to eat a sugar lump containing poliovirus, done developed by Dr. Sabine, and I was amazed. It's the nicest thing to be vaccinated, eat just a sugar lump. I don't know whether we'll manage with the SARS-CoV-2 vaccine. It's urgently needed, but this was, of course, been fantastic when we could do an oral vaccine. That would be the easiest, or an intranasal vaccine. That is my dream. And whenever I can contribute with our assays into this goal, I am happy to do so. So I want to give credit to a lot of people. There is, I told you, a lot of mobility and mobilization in scientists, but there is a lot of collaboration now. Everybody is open to share data, even unpublished one, as I did now in this talk. Most of our studies are not published right now. We got a lot of reagents from Katri de la Rosa and at MDC from Berlin. We got the spike vector from spike protein from Matthias Tenbusch in the Klaus Überla lab, and we got the hybridomas from Hans Martin-Jeck, and we thank them for this. We are getting Sierra from the medical hospital at the University of Freiburg, from Martin Schwemmler, Hartmut Hengel, and from Reinhard Voll. And we got EcoR expressing Ramos cells from my long-term collaborator, Jürgen Wienans and Niklas Engels. And so I want to also give a lot of credit to Yin-Yang Jing, who is a group leader in my group, and he's all the cloning and all the brainstorming I cannot do alone. I need him. And of course, I'm pleased to have a lot of collaboration with Julia Yulasova, who is an expert in metabolic signaling. And these are other people I grew up with. I'm very happy to work together with them. And we are funded by the DFG over the Transregio program and by BIOS. And at one point, I also had an ERC. Thank you for your attention.
Hi, Michael. Thank you. Thank you very much for your presentation. As you know, Rita had technical problems and could not join us for the Q&A. So if it's okay for you, you will find some questions in the Q&A chat. I don't know if you can reach them. And perhaps you can read some of them. Perhaps you can answer to the attendees some of these questions. That would be great.
Okay. Well, then I have to screen through this first.
Yes, of course. Just take a bit of your time for do that, and thank you for all attendees for your patience.
Okay, let's see. Most of Most of them only say hello.
Oh, no, you have to go to the Q&A chat. Q&A. Sorry, it's next to the chat.
Q&A. Okay, now I see
Yes.
it. Okay, and then let's see. Yeah, so the one question of Dr. Mohamed Farid, is the spike S protein same in antigenicity with other coronavirus? Well, I mean, the common coronavirus also has a spike, and it's a similar structure, but the sequence is actually quite different. So there is very poor cross-reactivity, but there is some, and therefore I think it will be very interesting when one can compare this in our assay in the flow cytometry side by side, which we plan to do. Then you can see directly whether many people have antibodies against, how many people have antibodies against these common corona cold viruses versus ones which have antibodies against the SARS-CoV-2, and what is the cross-reactivity. So that is our goal, right? Well, then the next question is, do you think one needs new vaccines every year like with influenza? And I think no. Because, I mean, as I told you, I give this lecture on Spanish flu and also on influenza every year. And of course, you have to know influenza is even worse in mutating because it has not only one genome, right? It has not only 130 kilodalton genome of RNA, it has 8. And therefore, you get all kinds of assortments between different influenza viruses, and that you don't get that with corona. But the bad news is, of course, both are RNA virus and have mutation rates. But it's the coronavirus, to my knowledge, is not changing as quickly as influenza. Therefore, I think we do not need every year a vaccine, but what we need, and that also was discussed by Andreas Radbuch, who's an expert on long-lived plasma cells, we probably need several boosts to get enough immunity against this virus. And then it will probably hold for a few years. Yeah, so the question about pH and pI changes in the R, whether that interacts is important things, so that we do not know, right? Of course, pH changes play more role not in the AR Golgi, but when the spike is taken up together with antibody or with the virus, or when rather the AC2 is taken up with the virus. Then, of course, it ends in endosomes, and the endosomes, as you all know, become acidic, and that is then enhancing the cleavage activity, and that, of course, plays a role. So another question is whether we can use the Ramos cells as a vaccine. So I said no. You know, these cells are, it's not a simple viral particle. A cell is a very complicated biological machinery and issue. It's a real life. There are thousands of proteins on the surface of Ramos cells, and you don't want to get immunized against them because, of course, partly they are your own surface. These are human cells. So no vaccine. These are analytical tools to understand the virus, the spike variability, and the humoral immunity against the spike. Or against other proteins. We can also express maybe the membrane protein on the surface of these Ramos cells. So are there any reports on monoclonal anti-treatment on clinical trials? Not to my knowledge, but there are, of course, reports where people use sera for infected people who developed antibodies. So this treatment is done. Right now, the monoclonal antibodies, I think, are not used yet in treatment. They're first tested in their activity. But this will probably enter the clinic in a few months. And there, of course, will be important to see what combination of monoclonal antibodies are best to use. And also here, I think our assay, our Ramos spike, SARS-CoV spike assay, may be interested in sorting out reactivity for which antibodies are blocking each other in binding to spike, which are helping each other, and which are blocking the binding site or not. So this could also be tested by other methods, but also with our Ramos spike assay. So that is the question, can the SARS-CoV-2 has access to other virions and then get maybe genomic, probably crossovers of genomics so that it can change? So I don't think so, because what I understand is that when you get a cold with common corona viruses, they are cleared by the immune system. They don't stay. It's not an HIV, right? Which you don't get rid easily of HIV once you have it, because the retrovirus is it's incorporated in your genome. Whenever you have a virus, you clear it. The immune system, particularly then finally the T-cell immune system, when it becomes active, will attack all cells producing viral protein and clear this virus from from our body. Yeah, so that is a question. So what would be the best way to admit a vaccine? So of course, one way is to inject it in the muscles, but it would be actually very interesting not to take it oral, as I said. I said a sugar lump would be great, but it would be indeed the nose. And the nose has a lot of lymphoid organs, tissues. And there it would be actually interesting to develop a vaccine which could be used to activate this immune system, which is the first attack site of the virus. Okay. So there is a question about when the virus will happen. So that is a big question. And I really not can make any give you any hints on this. So you know, Hans Martignac also referred to this, 130 different trials to develop the vaccine, and there is a big race right now. Well, how good they will working and when it will be there is not clear right now. Yeah, so sublingual vaccines we already mentioned. Another question, yes, that would be one way to go. Yeah, so there's another question, I don't know, TRM, that I don't understand what it means. So, sorry.
So, Michael.
Yeah. Yes, so there's one important question, so I don't want to keep you too long. There's one important question about why this antibody titer wanes so quickly. So it has to do with signaling B cells. And yeah, so that is actually a very important question. So why are the B cells not optimal activated to get into the long-lived memory B cell pool or in a plasma cell pool? And is there actually the virus producing? You know, viruses have a lot of tricks. Is the virus producing something which blocking this? And this would be actually interesting to figure out. So that is also my major question. So what are the bad tricks of the virus to prevent this efficient humoral immunity? Yeah. Okay. You had a question?
No, just whenever you want, you can just tell us whenever you are ready with the Q&A, and then I will jump to.
Yes. Right. Let me see where we are. So there was a technical question about how CRISPR works in Ramos B cells. I think there's no secret. We do the normal protocol. You know, you transiently express CRISPR-Cas9 with the vector and with the guide RNA, and then it works. So you can do this with many cell types. There's no special trick with Ramos cells. General protocols you find in many papers how to do this. So another question, are there other receptors for SARS-CoV-2? Yeah, so that is discussed, whether there are also sub-receptors. I do not know about this. So the high affinity receptor is clearly AC2. We see that also in our system. We have soluble AC2 gotten from Catarina Della Rosa, as I mentioned already, the name from Berlin. She produced the soluble AC2 that binds with high affinity to our spike expressing Ramos cells. But of course, when the other candidates are there, we can easily test this in our system. Yeah, so the other question is, what about T helper cells? You know, in real life, B cells do not grow when they see antigen. They get a first kick, and they need for prolonged growth and differentiation, they need T cell help, and that happens in the germinal center. And the Ramos cells are germinal center-derived cells, and they have learned to live without a T cell happily by changing their genome and being oncogenic transformed. But indeed, of course, when you're interested in vaccines and human immunity, you not only stare at B cells, but also you are looking at T helper cells. Yeah, so there is a comment that indeed there was a question about monoclonal antibodies, and there is already here mentioned that Regeneron has monoclonal antibodies in clinical testing, and I'm not surprised about it, because one of the founder of Regeneron worked next to me as a graduate student in New York, George Yancopoulos, and so I know him very well from this time, and I wish him all the luck that his company is producing rapidly this treatment, which is urgently needed for patients which have a bad cause of the COVID-19 disease. Yeah, but of course there are also other companies are doing this, and so there will be hopefully soon treatment for these kind of cases. So what is the sensitivity of the spike assay? And that is indeed, which we still have to figure out in detail. So I didn't show you the titrations and the comparison of the titrations done in comparison to other techniques. Of course, there are other spike assays. There's ELISA techniques. There are other methods to measure antibody interaction with an antigen. And that is the next thing we want to do. Actually, it's done today, the first assay by my master student, Nicolas Lesper. I mentioned him. He's doing this assay. So we do not know yet in comparison how well that assay works. So that has to come. So what I told you is ongoing research. So then it's a question, so do we have checked the B cells in COVID-19 patients by staining IgD and IgM? So that I like this question, because the first time somebody asked for IgD, which mostly, I told you mostly, yeah, so I do not know whether that has been studied. It's of course difficult to study in a patient in the ongoing disease, the B cells. And of course, very often the B cells, when they're activated, they switch to other isotypes like IgG. But and that cannot be easily studied. You know, you cannot take a pace. The B cells are mostly in the activated state. They happen in lymphoid organs in the germinal centers and not in the blood. So they're not accessible. But in our assays, we can, of course, generate B cell receptor for the spike, which are B cell IgM or IgD or IgG class, and see whether there's a difference in the sensing of the virus. Okay. So there was a question, what is your opinion about plasma therapy? Well, I think this is passive immunization, and this is established since a long time. Actually, in polio, I mentioned this epidemic. The first treatment was exactly that. So that passive is an old thing, you know, already as Hans-Martin Jeck pointed out, the first people, Van Behring and Kitasato, which worked on the discovery of the antibodies, then called antitoxin, they used a passive transfer of serum. So that is an established protocol. So it can work. How well it works in comparison to the new therapies may come up with monoclonal antibodies, one has to see. Yeah, so the question is, I heard the spike protein is mutating. Yes, of course. I told you it's an RNA virus, and when you look at the sequence of CoV1 versus CoV2, you find already a lot of changes. So one has to be prepared that the virus change, but it doesn't change in such a enormous rate like HIV, and that is, I think, therefore it's manageable. When man develops a vaccine, I think this vaccine will be for some time valid and can maybe confine this epidemic. Good. So the question is here, is this virus shed inside extracellular vesicles? I don't know. You know, I don't know what extracellular vesicles, there is also a phenomenon like exosomes, so that exosomes, vesicles from the inside get out. I don't know what the connection to exosomes is and virus, but what is clear is the virus is shed, it's produced in this ER Golgi compartment, and it is released by just the normal way how this vesicles from the Golgi are placed on the fused with the plasma membrane, and then it's released. So that is a normal way of this virus. Whether it actually stays also in other vesicles, it's not clear. Good. So I think I went through most of the questions. So there are patients, there are of course some questions which I think better a clinician than a molecular immunologist like me answers. For example, from Rashmi Singh, patients who survive after infection are getting the infection again as per reports. Yeah, so that is actually that is one of the question now. So what about these people who get a very mild course of disease? And you know that the virus makes very different diseases. It has a very mild course in many. You have a few fever just for a day or two, and then it's over, or it can have a very severe course by getting a pneumonia. And it's clear also from our studies, we see that actually also in our first assays. Sera from patients who went through a very long disease course, they have more antibodies in their sera than patients, or rather people who had just the very mild symptoms. So the question is, can the virus affect these people again? And that is possible, I think. But to what extent that happened, we don't know, because there is also the idea that many people are having mild colds because they have some mild immunity against this common cold coronavirus. And that saves them from being, can control the virus, at least so that it cannot amplify rapidly. And therefore, get the immune system more time to deal with it. Yeah.
Okay, Michael, time is running, and I think we are fine. I really would like to thank you in name from IOIS for your great webinar. It was really fantastic, and I think all the attendees think the same. So looking at the chat, I have seen that the comments were very positive. So I really would like to thank you for this great webinar.
Yeah. So I want to thank all my colleagues to being so cooperative, and of course all my students to be so curious about it. And I think for scientists it's a very exciting time, and yeah, we have to do something, and we all aim to do something, to learn more about it, and to maybe do something against it.
Yes.
And be beating the virus to pieces. Thank you.
Yes. Thank you very much. And I would like also to thank you to all attendees to be here, and hope to see you on the next webinar. You can have more information in ioas.org webinars. Next webinar will be on the 31st of July, and there you can find more information. So thank you again, Michael. Thank you to all attendees, and see you to the next webinar.
Okay.
Thank you.
Bye-bye.
Bye-bye. Bye-bye.